Hello everyone and welcome back to my channel. In this session, I'll be discussing about conjugation in bacteria. The types of conjugations, how they occur in bacteria. Let us try to understand. So, first of all, what is conjugation? Conjugation is a natural phenomenon taking place in bacterial cells where two cells are required and conjugation occurs between two cells. One cell is called as a donor cell and another cell is called as an acceptor cell or recipient cell. So the DNA from the donor cell will be passed on to the acceptor cell through a conjugation tube or a conjugation canal is formed. And finally the acceptor cells will also get the new DNA from the donor cell. Let us see what are all the different types of conjugations and what is the result of conjugation. Before we start actually the process of conjugation, let us try to understand about the donor cells. There are two types of donor cells. So, conjugation brief history, if we go into, it was first studied by Lederberg and Datum in E. coli. And E. coli is called as a lab rat of biology. It is called lab rat of biology because all microbiology, because all the microbiological studies are done in E. coli. Okay, now I will explain what are the two types of donors. So first type of donor called as F plus cell. This is one type of donor. So in F plus cell there will be, so this is the E. coli. It will have a normal its own DNA, its own genetic material. Apart from that, in addition to its own genetic material, it will also have an extra genetic material which is called as a plasmid DNA. All of you are aware of the plasmids present. So this extra genetic material, it is called as a plasmid DNA. Now, this plasmid DNA has its own advantages. It gives resistance to the bacterial cell against the antibiotics. Plus other whatever genes are present in plasmid, those characters are transferred to the bacterial cell. Now this plasmid has a gene which is called as a F factor. This F factor decides the sexuality of the bacterial cell. So the bacteria with this F factor, it is called F plus cell or a donor cell or a male cell. And another feature of this cell is this cell also develops a type of pili called as a sex pili. In bacterial cells there are pili which helps in attachment. And in addition to that there are some sex pili which help in process of conjugation. So this F plus cells will have a sex pili at certain places on the bacterial cell. At certain places. So wherever there is a sex pili present, that will induce the formation of conjugation tube. If so, whichever position it has sex pili, that results in formation of conjugation tube. That is the importance of sex pili. This sex pili will be hollow tubular structure. You have to imagine just like a spring. This will be present in the form of hollow tubular structure. So wherever there is a sex pili, this will induce the formation of a conjugation tube in the donor cells. So this is one type of donor in which only the plasmid will be transferred to the acceptor cell. Not the actual DNA, but the plasmid will get transferred to the bacterial cell. Now let us see the second type of donor called as HFR. HFR. HFR stands for high frequency of recombination. HFR stands for high frequency of recombination. That means here transfer takes place at a much higher rate compared to that of the F plus cell. So here the plasmid is present but it is not separated. It is not separately present. Instead, the plasmid which is present, it is completely integrated with the bacterial DNA. This plasmid DNA, it is completely integrated with bacterial DNA. And this DNA which is completely integrated, it is now called as, episome. It is called as a episome. So there is a difference between plasmid and episome. And episome will be completely integrated. Episome will have, this side we can call it as a ending point, and suppose this side it is a starting point. The F factor may be present anywhere. Normally F factor in these episomes are present almost towards the end. So the conjugation will start wherever there is a starting point. So here the advantage of this is it shows high frequency of recombination. Much faster the DNA transfer can take place. So this is another second type of donor cell called as HFR. Now let us try to understand the two types of conjugations. So first I will start with one type which is called as F plus F minus conjugation. Let us try to understand. Then we will do the other one. So in the first type, let us try to understand. First type of conjugation, that is F plus and F minus conjugation. F plus and F minus conjugation. So I told you F plus is the donor cell, F minus is the recipient cell. So recipient cell will not have any sex pili and it will not have any plasmid. It will have its own DNA. So suppose this is the F plus cell. You know F plus cell will have its own DNA. Along with that, it is going to have a plasmid DNA also. And conjugation tube is formed between F plus and F minus cell. This is called as a conjugation canal or conjugation tube. And F minus is going to have its own DNA without the plasmid. Now once the conjugation process starts, then this plasmid will get transferred to the F minus cell. First the plasmid becomes single stranded. It becomes linear first. That is, at one point it breaks and it becomes linear. And after it becomes linear, one of the strand will be transferred to the recipient cell through a conjugation tube. One of the strand will be transferred. After one strand is transferred, we can get the result as, so this is the normal DNA present, and each cell will have one strand of plasmid. After one strand is transferred, each cell is going to have only one strand of plasmid. This condition of the cell is called as hemizygous condition. This condition is called as hemizygous condition. Now next stage what happens is, the other strand will be synthesized in both the cells. It is single stranded, so it becomes double stranded after DNA replication. So after the conjugation, at the end of the conjugation, what will happen? The plasmid DNA in both the cells will become double stranded. Plasmid DNA can become double stranded. So this F minus cell at the end of conjugation, it is converted into F plus cell. This is very, very important feature in this type of conjugation. So what happens at the end of conjugation? At the end of conjugation, all F minus cells are converted into F plus cells. This is very important feature in this type of conjugation, and this is most common type of conjugation. Very commonly it occurs. Now let us try to understand the second type of conjugation, that is called as HFR and F minus conjugation. The second type of conjugation is called HFR and F minus type of conjugation. Here already, as we discussed, HFR stands for high frequency of recombination, that is one, and here plasmid is not present in separated state. It is present completely integrated with bacterial DNA, and that integrated state of plasmid, that is called as a episome. So here what happens in this type of conjugation, say this is the donor cell and acceptor cell, this we call it as HFR and F minus. So here we find the plasmid DNA is completely integrated. Now say one is the starting point and say other is the ending point. Suppose this is the starting point and this side say it is the end point. So always conjugation or transfer takes place from this point. It starts from this point. So here also first the DNA becomes linear. It breaks at this point. What happens in first stage? DNA becomes linear. DNA becomes linear. After the DNA becomes linear, DNA is transferred. DNA is transferred from starting point. DNA is transferred from starting point. Wherever there is a starting point, DNA will be transferred. But complete DNA transfer cannot take place because conjugation time is less than that of complete DNA transfer. It takes more time for complete DNA transfer, whereas conjugation time is much less. So conjugation lasts for less time compared to DNA transfer. What is the other important point? Conjugation lasts for less time than DNA transfer. So as a result, what happens? Only some part of DNA will be transferred, and F factor also may be located at the end. So when F factor is located in the end, there is almost all the time there is no transfer of F plasmid. So conjugation lasts than DNA transfer, and F factor is rarely transferred. F factor is rarely transferred, but just some amount of DNA can be transferred. Some amount of DNA. So the recipient cell initially is having only DNA. So this recipient cell now can get only some amount of DNA, but complete transfer will not take place. Complete transfer cannot take place. So therefore, since complete transfer is not taking place, the recipient cells will remain as F minus only. They are not going to get converted into F plus. Unlike we find in this conjugation F plus and F minus, where all acceptor or recipient cells, they are converted into F minus. But here they are not getting converted into F minus because we find only very little DNA can be transferred. But since it takes place with a very high rate of recombination, very high rate, it is called as a high frequency of recombination. So in the end, what can we write is all F minus cells are not converted. All F minus cells are not converted into F plus cells. All F minus cells, they are not converted into F plus cells. But they receive some DNA, some genetic material from the donor cell. And this type of conjugation is called as a true conjugation, because there is actually transfer of DNA taking place. If this is the starting point, the bacterial DNA will also get transferred. So HFR actually can be called as it is called as true conjugation. True conjugation. Whereas F plus and F minus cannot be called as true conjugation. It is not true conjugation. Not true conjugation. This is what we find difference between HFR and F minus conjugation and F plus and F minus conjugation. Conjugation is most common process occurring in nature. We have studied about other two types also, transformation and transduction. Transformation occurs very, very rarely in nature, unless if there is cell is exposed to any extreme conditions. Then transformation can occur. Transduction occurs in nature, but it is again very rare. So in nature, the most common type of the transfer reproduction which occurs is conjugation. Again, there is another very important point regarding sexual reproduction in bacteria is it is not a true method of sexual reproduction. It is not a true method of sexual reproduction because there are no sex organs formed or there are no gametes formed, and there is no gametic fusion also. So because of this, there is no true sexual reproduction in bacteria. But still we call it as sexual reproduction because there is a transfer of genetic material taking place. There is a transfer of DNA taking place. So therefore we still call it as a sexual method of reproduction. Okay, with this I complete the process of conjugation. In the end, I want to discuss a small one numerical which is associated with this type of conjugation. Okay, so I am going to give one numerical based on mainly F plus and F minus conjugation. So first I will write that question here. So the question says if there are 75 F plus and about 50 F minus cells, 75 F plus and 50 F minus cells, and after the conjugation, at the end of conjugation, if conjugation is taking place between all the cells. So question is if conjugation occurs in all cells, how many F plus and F minus cells are present at the end? How many F plus and F minus cells are present at the end? So once again I will read. If there are 75 plus and 50 minus cells, if conjugation is occurring in all cells, how many F plus F minus cells are present at the end? So in the first stage of conjugation, we find out of 75, 50 plus are taking part. So 50 plus and conjugation between 50 F minus. So what is the result? All 50 F minus are getting converted into F plus. So at the end of this conjugation, we get a total of 100 F plus cells. If conjugation is taking place once, at the end we are going to get 100 F plus cells. How many are remaining? Out of 75 we have taken 50. So 25 F plus are remaining. They cannot undergo any conjugation. So 25 F plus are remaining. They are not going to undergo any conjugation because all are F plus. So total how many F plus are present? You are going to add this 25 F plus. That is equal to total of 125 F plus cells will be present at the end of conjugation. This is a very common, very simple question, can be expected. So that's all for now, all my students and all my subscribers. So I tried to discuss the process of conjugation in detail. So in my next video, I am going to take up another topic from the same microbiology and I am going to discuss. So thank you very much. Have a good day and please do subscribe and share to the maximum extent. And if any queries, anything, you can definitely ask and write in the comment box. So thank you very much. Have a good day.